Vitamins and minerals, biotin. Biotin is going to be the next water-soluble B vitamin that we were going to discuss that's involved in reactions of energy metabolism. We see here the structure of biotin and the different components. We see this stacked ring structure, including a urea ring, a thiophene ring, and this valeric acid side chain, so altogether making up the biotin structure. We see food sources of biotin, so food sources that will be high in biotin is going to include liver, soybeans, egg yolk, cereals, legumes, and nuts. We also see that biotin is made by bacteria in the large intestine. We also see, in terms of the bioavailability of biotin, implications of this glycoprotein called avidin. So avidin is found in egg whites. We see here that egg yolks contain biotin. However, when consuming a raw egg that has, again, both the egg white containing avidin and the egg yolk containing biotin, the avidin, this glycoprotein in the egg white, binds to biotin. So avidin binds to biotin in the egg yolk, making it unavailable for absorption. Therefore, it's important to cook eggs, especially the egg white, to limit the activity of the avidin and decrease its binding to biotin. So just to keep in mind in terms of the bioavailability of biotin in egg yolks, if consuming eggs raw, then it may not be bioavailable due to avidin. So most biotin is consumed in the protein-bound form. So since we absorb free biotin, the protein-bound biotin must be hydrolyzed to free the protein-bound biotin to the free form. So the digestion of biotin is occurring through proteolytic enzymes, and so the digestion by these proteolytic enzymes produces different structures. So we get the free biotin, but we also get biocytin and biotinopeptides, which need to undergo further digestion. So first we have digestion by proteolytic enzymes, which does yield some free biotin, but the biocytin will need to be further hydrolyzed by the biotinidase enzyme, and then the biotinopeptides will need to be further hydrolyzed by additional proteases. So once we are able to fully digest biotin to the free form, we have the absorption of biotin, and the absorption of biotin from food sources will be absorbed further down in the small intestine, so in the jejunum and the ileum. Biotin synthesized by gut bacteria in the large intestine will be absorbed in the proximal colon. So in terms of our relevant dietary or exogenous sources of biotin, the bacteria is going to make up a small proportion of that. Most of the biotin that we will be absorbing will be from food sources. We see different mechanisms of absorption. So at physiologic doses, we see absorption through this SMVT carrier through the small intestine in the jejunum and the ileum, so the brush border of the enterocytes in the jejunum and the ileum. We saw previously that the SMVT carrier is also used to absorb pantothenic acid, so we're going to see that also occurring with biotin. Pharmacologic doses, so very high doses of biotin, will see absorption through passive diffusion, but the primary method of absorption is going to be through this SMVT, sodium-dependent carrier. Once biotin has been absorbed in the enterocyte, then it exits the enterocyte through the basolateral membrane into the bloodstream. Once circulating in the bloodstream, it's going to be transported freely as free biotin, so that's biotin. So that most biotin in circulation is going to be in the free form. However, some will be bound to the protein albumin. When discussing biotin functions, it's going to be acting primarily as a coenzyme. A coenzyme for particularly what are called carboxylation reactions. So these carboxylation reactions will be reactions where we add a carbon, particularly in the form of CO2. And this is going to be in contrast to decarboxylation reactions, which would be the removal of carbon in the form of CO2. So we see decarboxylation reactions will be the conversion of pyruvate to acetyl-CoA in the pyruvate dehydrogenase complex. So in that reaction, we remove a CO2. However, a carboxylation reaction would be, for example, conversion of pyruvate to oxaloacetate. In that reaction, from pyruvate, which is 3 carbons, to oxaloacetate, which is 4 carbons, we add a carbon in the form of CO2. So there's a number of steps, or there's several steps here involved with activating and preparing the biotin-dependent enzyme for its activity. So you can see here that the reactants and kind of the components of this will need to be first activated. So activated in this energy-dependent reaction that includes ATP and bicarbonate. So with this ATP and bicarbonate, with this reaction, we see the formation of this carbonic, phosphoric anhydride. So this is going to be active with this phosphate group attached, and then we see this CO2. And then we see the biotin enzyme. So this is the biotin structure, and then in this active site here, it binds to the enzyme that it's going to be working with. And then in the next step, we see the addition of the CO2 to the second active site. And so we see removal of the phosphate group and then binding. So again, binding of the CO2 to the biotin enzyme, forming this carboxybiotin enzyme, including biotin, the enzyme, and the CO2, which is now ready for the carboxylation reaction. We now see the list of these biotin-dependent enzymes. So there's 4 key biotin-dependent enzymes that we'll discuss. So first, pyruvate carboxylase, converting pyruvate to oxaloacetate. So that's that example that I had mentioned previously. So pyruvate being 3 carbons, oxaloacetate being 4 carbons. And with this carboxylation reaction, we add this carbon in the form of CO2 to pyruvate, making oxaloacetate. So the significance of this, it replenishes oxaloacetate for the TCA cycle. So pyruvate converted to oxaloacetate allows for adequate oxaloacetate for Krebs cycle activity. Also, pyruvate carboxylation is needed for the process of gluconeogenesis. The second biotin-dependent enzyme that performs these carboxylation reactions is called acetyl-CoA carboxylase. So acetyl-CoA carboxylase, we mentioned previously, is a key enzyme, actually the rate-limiting enzyme of fatty acid synthesis. And so in this reaction, we have acetyl-CoA forming malonyl-CoA, and we see that it is the priming step, the preliminary step to the fatty acid synthesis. The next enzyme that we're going to discuss is propionyl-CoA carboxylase. We've seen propionyl-CoA as a product of the oxidation of odd-numbered fatty acid chains, and we also have discussed that propionyl-CoA then can enter the Krebs cycle. So propionyl-CoA enters the Krebs cycle as succinyl-CoA, but prior to being formed to succinyl-CoA, propionyl-CoA is converted to what's called methylmalonyl-CoA. So this propionyl-CoA carboxylase converts propionyl-CoA to methylmalonyl-CoA, which then can form succinyl-CoA. And we saw again here that this provides mechanism for metabolism of some amino acids and also odd-numbered fatty acid chains. So propionyl-CoA is catabolism product also of some amino acids. The last enzyme, and we haven't seen this enzyme or its intermediate previously, however, we see this beta-methylcrotonyl-CoA carboxylase involved particularly in the catabolism of the amino acid leucine. So we're going to see a diagram here that's going to be highlighting a number of these reactions where we see biotin involved. So first here we see biotin as a coenzyme for the conversion of this propionyl-CoA to methylmalonyl-CoA, and we see that that's going to allow for eventual conversion to succinyl-CoA and oxidation through the Krebs cycle. Next we see biotin here in the process of fatty acid synthesis. So biotin is involved as a coenzyme of that acetyl-CoA carboxylase. And then lastly, we see biotin here involved in the conversion of pyruvate to oxaloacetate. So we have biotin as a coenzyme to the pyruvate carboxylase enzyme needed to convert pyruvate to oxaloacetate. Here's again a diagram with kind of further detail of this biotin dependent enzyme. So we saw the template basically of these, the carboxy biotin carboxylases, but we see the specific biotin dependent enzyme here. So we see the biotin structure, we see the pyruvate carboxylase, and we see the CO2 here. And then we see with pyruvate, which is 3 carbons structure, we see that the CO2 is going to be added to the pyruvate to form oxaloacetate. And we also see with many of these reactions, they're going to be ATP dependent. So we see ATP involved, and then magnesium also being involved as a cofactor. Here we see further detail with the conversion of propional-CoA to D-methylmalonyl-CoA. We see propional-CoA is going to be formed from the oxidation of odd-numbered fatty acids chains, and then also from the catabolism of certain amino acids, so threonine, methionine, and isoleucine. So in the conversion of this propional-CoA to the D-methylmalonyl-CoA, we see biotin involved as coenzyme for this propional-CoA carboxylase. We also see, like with the last slide, the investment of an ATP and the involvement of magnesium. Lastly, here we can see the catabolism of leucine. We see its deamination and conversion to the beta-methylcrotonyl-CoA, and the activity of the beta-methylcrotonyl-CoA carboxylase. We see that it's the biotin dependent. We see involvement of ATP and also magnesium, and then the eventual conversion to its catabolism products, which we have discussed previously, being the acetoacetate and acetyl-CoA. Next, we'll discuss the adequate intake level of biotin. So notice that this is not an RDA. We have the AI intake level for biotin. For adults and also in pregnancy, the AI level is 30 micrograms, so it's actually a very small amount that is needed daily. It's a little bit higher in lactation. So deficiency is rare, but it can be serious, leading to lethargy, depression, alopecia, which is hair loss, nausea, and anorexia. So this is not common, but it could pose some serious risks